Hi, my name is Sanchari Ghosh, and I'm the content specialist at bioRxiv, Cold Spring Harbor Laboratory. And I'm here today talking to Professor Anne Brunet. She's the Michelle and Timothy Baraket Professor of Genetics at Stanford University, where she studies molecular mechanisms involved in aging and longevity. She's had a very exciting career speckled with several very prestigious awards, which include the NARSAD New Investigator Award in 2009, the Pioneer Award of NIH Directors Fund in 2012, and most recently, the Bennett J. Cohen Award for Research in Aging, University of Michigan in 2014. Thank you, Anne, for taking the time to talk to us today.
Thank you so much.
So one of the most interesting aspects of your research in your lab is the fact that you use a lot of different organisms, right? And one of them is not the most common, the African killifish. So would you spare a couple minutes and tell us what makes that an interesting organism?
Yeah, so we were excited by the African killifish because, like the aging field had strongly benefited from short-lived invertebrate models like the
Right.
worm, also the Drosophila, and also the non-vertebrate like the yeast to make key discoveries of aging. And then in parallel, the field had also strongly benefited from mice and studies in mice and cells in mice and humans, but those take much longer.
Longer
And
to age.
yeah, longer to do. Mouse lives about 3 years,
Yeah.
and so that's much longer. And then the non-vertebrate are terrific, and they've revealed really like pathways that were conserved, but they are also missing some key elements that are only present in vertebrates. So they are missing, for example, an adaptive immune system like T and B cells, and those are critical during the aging process in humans. They're also missing some aspect of stem cell biology in a lot of tissues. They have some, but not all of them, or like cardiovascular system, etc. So this is why we thought like, oh, this African killifish, because it's a fish, it's a vertebrate, so it has all those key tissues, but it lives on a scale that's compatible with high throughput. It's scalable.
Right.
High throughput experiments.
Right. So it's a very convenient model system to do kind of best of both worlds. I see. That's great. But the work you presented at the symposium last week was based solely on mouse neural stem cells, right? Yes. And before we go further, could you help us clarify how are these niches, the stem cell niches that you work on, how are they identified?
Oh, so they were identified a long time ago, based on markers of proliferation. For a long time, actually, like the brain was thought to be entirely post-mitotic and just having like differentiated cells. But from very time from Francisco Nottebohm, Alejandro, Arturo Alvarez Bulia, sorry, and then also Fred Gage from the other niche, like they were able to identify stem cells in so cells that are still able to proliferate in the adult brain, in birds, in mice, and also in humans. It's been a little bit controversial in humans over the years, but there is a consensus that there are for sure stem cell or cells with the potential to be stem cells in human brains as well. So yeah, that's so those cells have been identified
markers
a long time ago. Yeah.
Okay, and so there were markers that you could use to identify them and extract them.
Yeah, so that's also the work, from Fiona Deutsch in Arturo Alvarez Bulia's lab. So the cells, not only they can proliferate, so that's one of the marker, actually, like the ability to incorporate an analog of a base, EDU, for example, or BRDU. But you can also use a combination, I would say, of markers. And initially, in the first types of studies we use fluorescence activating cell sorting, so FACS. With a combination of different markers to purify, freshly isolate those cells from the brain. But more
Right.
recently, with the single cell, the advent of single cell technology, you
Yeah.
don't actually have to purify based on
Right.
markers. You can take the entire
Right.
tissue and they will be there, I guess. Yeah.
Okay. So that was going to be my next question. So you have moved on from the previous paradigm of bulk data onto single cell data, right? So what are the advantages apart from the fact that you don't have to sort so carefully?
Yeah, so the advantage of the single cell technology is that yes, you don't have to necessarily have markers. So sometimes it's really helpful when cells, it reveals cell heterogeneity, maybe cell state that weren't known, or different cell transitions that weren't known. It's also helpful for cells for which the markers are a little bit ambiguous. And then, because it doesn't depend on markers, for example, during aging, if the markers change slightly, that might
Right.
affect the results of fluorescence activity in cell sorting, whereas here it's more quote unquote unbiased. It's never
Right.
truly completely unbiased, but it's more
Fair.
unbiased.
But it also allows you to capture a continuum, a continuous spectrum of marker expression, right?
Yes. So then you
Okay.
can, in fact, we see that very neatly. I don't know, like if you were like in the talk I presented what's called
Right.
the Disney or UMAP plot, like a representation plot, a
Yep.
representation in 2 dimension, and you can really see that, especially the lineage of the stem cells, they form this kind of neat continuum where
Right.
you
Right.
see the intermediary step, but you also see the different cell types. That was really cool to see, actually.
Right. So do you expect to see similar cellular clocks in other organisms, single cell clocks in other organs as well as organisms? Are you looking for them?
Yeah, so we have this. It's I guess unpublished, but we have this gigantic terrific collaboration with Tom Rando and Peggy Godell as part of a program project funded by National Institute of Aging on aging, and there, the goal is to compare from the same animal the
Mm-hmm.
brain stem cells, the muscle stem cells, and the blood stem cells
Right.
in
Okay.
response, in this case, to exercise. So that it's been
Right.
cool to analyze the data and see differences and similarities between the different tissues.
Nice. Exciting. And then you move on to look at two extremely interesting interventions to sort of push back the clock. And the obvious question that comes to my mind is why parabiosis and not just a simple transfusion?
Yes, so the simple transfusion of blood, the way it's been done, it's true that it recapitulates at least some of the effect of parabiosis, but it's never as potent as parabiosis, number one. And then number two, the way it's done is that you have to collect the plasma, of
Right.
course, from a young animal. And the
Right.
mice have very little plasma. So in the end,
Uh-huh.
one needs a lot of mice to basically
Right.
collect and store the plasma and then inject repetitively every week and etc. So we felt that for the first pass experiment, the parabiosis, because it's been used so much
Done.
by so many groups, and in this case you let the biology do the exchange for you, and the
Right.
exchange is done constantly by the biology of the organism, was a better proof of principle to start with.
Right.
But I
Right.
agree with you that eventually, especially to isolate, I mean, parabiosis, it puts mice. I got the question actually, like, oh, it does many more things than, just transferring the blood factors. Of course, there's the blood cells. One can imagine oxygenation is different. Someone suggested in the Q&A that, in fact, the mice were kind of like circadian together. So maybe if the young
Yeah.
sleeps, they could then train each other in other ways as well.
Right.
Right? So,
Right. Right.
yeah,
Right. Yeah. Okay. So, but like exciting things to look forward to, right?
absolutely.
Um, yeah, and very exciting data, like the the specificity that shows up in the genes that are modulated, remarkable. I'm just curious, do you expect the changes to be more acute, or more chronic and long lasting? And is that expectation the same for parabiosis versus exercise?
Yeah, so it's been shown by groups that have been working with parabiosis. I know less about exercise, but but I'm pretty sure it's similar. With parabiosis, that if you detach the mice and then look at them, let's say month after you've detached them, that part of the a large part of the benefits are actually lost, and it would need
Mhm.
to be a little bit more continuous to keep some of the benefits. It's kind of like a
Uh-huh.
transient benefit. And same for exercise. I think it's probably less directly studied, but everyone who's done exercise probably knows that you need to continue doing
You
it.
have to keep doing it.
Yeah.
Right, right.
So yeah, so there is probably some, one could use the clock actually to determine, because then it's more quantitative. So one
Right?
could use it to
Exactly.
determine the portion. There's probably a memory of
Hmm.
exercise and parabiosis, but it's probably also a large part of it is erased, and it'd be interesting to, at the cell type level and at the gene level, being able to
Hmm.
separate the cells and the genes that may keep the memory for longer. So
Hmm.
yeah, that the clock could be actually also very put to use for that aspect. That's yeah, we hadn't thought of that, but thanks for the suggestion. This is cool.
Well, um, the other thing I was wondering is, given that the gene sets that are modulated by these two different interventions are fairly separate, um, how how are you thinking about it? Like, do you think different interventions can have synergistic or antagonistic effects, and can that be predicted from the gene signatures?
Yeah, so one idea we haven't done that yet, but was to use the almost like the the modeling power of having all these data to create almost like vectors to sort of like
Mhm.
see in which direction almost like the different rejuvenation intervention go, and try to see whether they could be combined, or maybe they might be antagonistic with one another. We think that exercise and parabiosis could be quite synergistic rather than antagonistic. And the reason we think that is because, but you never know. The reason we think it's synergistic is because we have noticed that exercise was producing some aspect of inflammation, actually, which aging
Ah,
does as well, but that
right.
parabiosis was good at removing that. So if
Mm,
the exercise induced inflammation has perhaps negative consequences to blunt some effect of exercise on
right.
health span, combining it perhaps with young blood kind of like
Yeah.
achieves like a synergistic aspect. But you never know, like it could be that the inflammation of exercise is actually needed for it to be
Mhm.
better. Also, like the circulating factor of exercise might be diluted if you have young blood coming from a non-exercised mice. So yeah,
Right.
it's quite interesting and fascinating to start thinking about the complexity of having
Yeah, yeah.
intervention.
Especially given that some of the genes are very tissue specific,
Mhm.
cell type specific, but some are fairly general. So I'm wondering how the whole different organ systems are going to work out. So that I think that'll be very exciting
Yeah,
to
yeah,
see.
exactly,
Um,
exactly. And you're right. Like, in fact, it's beyond the cell type. It also could be that some
right,
organs respond
right.
better than others, regardless
Right.
of cells. Yeah, absolutely.
Which kind of brings me to the next question is what do you think will happen to the clock in cases of diseases? Like, I know that one of the focus on this is because of degenerative diseases that set in with aging, but what about like organ specific diseases? Do you think the clock on one organ will kind of show the disease while the others will remain like more true to the chronological age of the organism?
Yeah, that's a great question. And I think Steve Horvath, who's been pioneering the clock with the DNA
Mhm.
methylation clock on organs and tissues, has been already like having data on aspects of
Mhm.
this. And for example, what he's found is that some disease accelerate the clock, like they act almost as like pro-aging types
Right.
of features. It seems to be quite general from what I remember from his studies, but you're
Mhm.
absolutely right. Like, what would be very interesting to test are the segmental progeria, for
Right.
example, that touch like
Right.
one organ compared to the other and see whether they are pro-aging more in the organ of that they
Mhm.
affect, or if they could be also systemic factors. I mean, that seems to be
Right.
also like a, you know, inflammation, for example, is also appearing,
Mhm.
and the immune
Right.
system is
Right.
disrupted with those diseases. So maybe even though they primarily touch a tissue, they might also
Right.
accelerate the clock at a distance in other tissue. But yeah.
Right. Yeah. Yeah. No, those are extremely exciting possibilities. And I'm like super excited about what comes forward from your lab. This is really good. Thank you so much for taking the time to chat with us. And this was very
Yeah.
interesting.
Thank you so much, Shari. Thank you. Great questions. Terrific.
Well,
Thanks.
thanks.